Hello to everybody and welcome to lecture 4 of this international course. The subtitle of my talk is Winter is Coming, and we will talk about the principle and practice of cryopreservation of reproductive cells and tissues in assisted reproduction. And writers of science fiction have been greatly attracted by the possibility to suspend the biochemistry of life and then revive it by cryopreservation of whole human bodies. But although there are some companies that offers the possibility to be cryopreserved immediately after death, indeed we are not able to preserve a human being, a whole body of a human being. And the case into patients in which amputated fingers were successfully cryopreserved and reimplanted represent the maximum size of body parts successfully cryopreserved. And but however, we developed throughout the years the ability to successfully cryopreserve cells and small-sized tissues. And now cryopreservation in assisted reproduction is a routine, and it is indispensable to cryopreserve sperm, embryos at their various developmental stage till the blastocyst stage, and oocytes, and even gonadal tissues. And this pioneering paper of a father of cryobiology, especially in the field of reproduction, this pioneering paper of Peter Mazur in 1963 put the basis of slow cooling, or the so-called equilibrium cooling. In fact, Mazur observed that when you cool cells into a medium, the medium becomes to be frozen, and solute concentration in the unfrozen part of the medium increase, and this drive a cell dehydration. And this is very important because according to the cooling rate, if we cool the cells too rapidly, the cell will not have the time to lose water, and intracellular ice will form, and the cell will retain the same volume but will not survive to cryopreservation. Instead, if we cool a cell at its optimal cooling rate, we will give the time to the cell to dehydrate, and cell volume decrease, and the protoplasm achieve a very viscous condition, and the formation of intracellular ice is avoided, and the protoplasm achieves a glassy, vitrified state. So, but about 14 years before, we see the publication of a pioneering paper in which the Parks group, Paul, Smith, and Parks, claimed to have successfully vitrified full sperm, and they were trying to cryopreserve full sperm using fructose as an extracellular cryoprotectant. So fructose increased the osmolarity of external medium, and this drive a cell dehydration. But they were astonished to see that the sperm were indeed motile after thawing, because they had previous experience with the fructose cryopreservation of false semen, and this was not successful. But anyway, they were not able to reproduce such findings until they understood to have accidentally exchanged the label of a fructose bottle with the glycerol bottle. So they used an intracellular cryoprotectant, glycerol. And the action of an intracellular cryoprotectant is that it permeates through the plasma membrane and substitutes cell water, so dehydrate the cell and depress the protoplasm freezing point. It is also important to observe that Polge used different concentration of glycerol. So the optimal concentration was 15%, but if he used concentration below 10%, the cells do not survive after thawing, and this was due to the formation of intracellular ice crystals. On the other hand, concentration of glycerol above 20% resulted in immotile sperm, and this was due indeed to the solute effects, and we will see this in the next slides. And another important observation made by Polge was that other intracellular cryoprotectant like propylene glycol and ethylene glycol were not effective because they exerted a toxicity on full sperm. But the Parks group was indeed lucky in another respect, because they were also working on rabbit sperm, and would they have applied the glycerol cryopreservation to rabbit sperm, it would not have been successful, because any cell type and each species have a preferred cooling rate, but also preferred cryoprotectants. And so the Parks group indeed described what is now called the slow cooling or equilibrium cooling. It is based, as I told you, on the formation of ice in the external medium that dehydrate the cell and avoid intracellular ice formation. So indeed, during slow cooling, there is extracellular ice, but no intracellular ice. So the cytoplasm is vitrified indeed. But at that time, freezing injury was thought to be due to ice crystals that pierced or teased apart the cells and intracellular structures and destroyed them through a direct mechanical action. But it was soon recognized that there were other causes of injuries, like the solute effect. In fact, in this pioneering paper of Lovelock in 1953, he exposed the red blood cells to the predicted sodium chloride concentrations achieved during freezing and thawing, and he obtained the same rate of hemolysis. And so he concluded that the high concentration of extracellular but also intracellular solute concentration during cell dehydration were toxic, and this effect was called the solute effect or the solution effect. And this was also the basis that pushed some authors to substitute, to replace sodium in the freezing medium with choline, because the cell membrane is not permeant for choline, and so it is avoided the formation of a high sodium concentration, both in the external but especially in the internal milieu. And this was successful for cryopreservation of mouse oocytes, but also of human oocytes, and produced the birth of 2 babies at that time. And we also applied the replacement of sodium with choline in slow cooling media to improve human ovarian tissue cryopreservation. But although there are other strategies of cryopreservation that are being explored in research, like directional freezing or directional vitrification, indeed, in the clinics, we apply two different cryopreservation strategies. The first is called, as I told you, slow or equilibrium cooling, equilibrium freezing. And it is based on a long exposure to low concentration of intracellular cryoprotectant, and typically what is used is propanediol and the extracellular cryoprotectant sucrose. It involves the use of programmable freezer, and so it is an automatic procedure. On the other hand, we can use vitrification. This is based on the brief exposure of cells to high intracellular cryoprotectant concentration. And typically we use the combination of two cryoprotectant, intracellular cryoprotectant, like ethylene glycol and dimethyl sulfoxide. And this strategy, the use of two instead of one intracellular cryoprotectant, is based on the concept that the specific toxicity of each intracellular cryoprotectant is lowered in this way. And vitrification requires very high cooling rates, and these cooling rates are achieved through the concept of minimum volume cooling, in which we load a very thin and conductive vitrification devices with, for example, just one oocyte covered just by a film or medium, and these, of course, increase the cooling rates. Vitrification, as we will see, is successful in assisted reproduction, is a successful strategy, but it is a manual strategy, and so it is highly dependent on the operator skill. And so, just to summarize, as regards slow cooling, if we cool a cell too slowly, we will get a solution injury and the cells will not survive a thawing. On the other hand, if we cool cells too rapidly, we will get intracellular ice injuries, and so each cell require an optimal cooling rate. If we use much higher cooling rates, we can vitrify the cells. But there is also another cause of damage that occurs during thawing or during warming. So in the case during cooling, we have the formation of ice nucleation or ice crystals. They can regrow to a larger size that is, of course, more damaging during thawing or during warming. This phenomenon is called recrystallization. And so, although slow cooling is the first procedure developed in cryobiology, it is being gradually but also rapidly replaced by vitrification. And let's have a look to a typical slow cooling procedure that was used years ago for oocyte cryopreservation. So oocytes are first exposed to 1.5 molar propanediol as an intracellular cryoprotectant, then to propanediol plus sucrose, and during this time at room temperature, propanediol substitute the cell water and drive an efflux of water, and the sucrose does the same, but in a different way, because it increases the osmolarity of external medium and so drives a cell dehydration. Then we cool to -8 the sample at a rate of 2 degrees per minute. During this time, the permeation of propanediol contains the dehydration of the cell cytoplasm. Then we hold the temperature at -8 for a time, and during this time, an important procedure is done. It is called seeding. What is seeding? Seeding is the controlled induction of ice formation in the extracellular medium. How we seed? We just touch the meniscus of the cryopreservant medium in which cells are immersed with the cold metal rod. And so ice begin to form, solute concentration increases, and so during seeding there is an essential dehydration of the cell. Then we cool very slowly at 0.3 degree per minute the sample to -30 degrees. And during this time, there is a slow dehydration and ice formation will not occur. Then we apply a faster cooling rate of 50 degrees per minute and cool the sample to -150 degrees. And during this time, the speed of cooling avoid the formation of intracellular ice. Then the samples are immersed, are plunged in liquid nitrogen. So slow cooling depends on freezing outside the cells and the absence of ice formation inside the cell. Each cell type has an optimal rate of cooling, and this depends on the particular cryoprotectants used and on the water permeability of the plasma membrane to both water and intracellular cryoprotectant. And recrystallization, as I told you, is the conversion of a large number of small ice crystals that are relatively innocuous into a smaller number of larger but more damaging ice crystals. And both during cooling and warming, we have traversed a dangerous temperature range from 0 to -120, 130 degrees, in which ice formation and nucleation can occur. And we can enhance the speed of cooling, but it is difficult to enhance the speed of warming because we are limited by the physiological temperature of the given organism. And so, as you see in these cooling and warming curves, the time spent by the sample in this dangerous temperature range is 3 times higher during warming compared to cooling. And this has been elegantly demonstrated by Mazur in more recent papers in which he demonstrated that the warming rate is dominant over the cooling rate, and the proportion is needed between them, with the higher warming rates compared to the cooling rates. And in fact, he was able to obtain a high survival of mass of ice after cooling at from very low speed, for example, 95 degrees per minute to 70,000 degrees per minute, provided that the warming was done at relatively higher speed, so from 600 degrees per minute to about 120,000 degrees per minute. And let's talk about a bit more about the cryopreservation injuries. So besides the physical damage of extracellular, intracellular, and intracellular ice formation, and in which most scientists believe that no ice formation occurs, whereas other scientists, on the basis of indirect data, state that if less than 4% of intracellular water will be converted into ice, the cell can survive to cryopreservation. And besides also the solution effect, there are injuries caused by the cold itself, so they are called chilling injuries. And there is also the toxicity of cryoprotectant, especially intracellular cryoprotectant. And so the properties of the plasma membrane are fundamental. For example, in oocytes and early embryos, both intracellular cryoprotectant and water traverse the membrane very slowly through simple diffusion across the lipid bilayer. Instead, at later stages, such as the morula and blastocyst stage, there is a substantial expression of aquaporins, and this drives a much faster efflux of water through aquaporins and the permeation of cryoprotectants through the same channels. And as concerned the oocyte cryopreservation, indeed, the first babies generated from embryos generated in turn by cryopreserved slow-cooled oocytes were reported in 1986 and 1987. But indeed, this procedure was largely unsuccessful. And in fact, it was calculated that about 100 oocytes were required to achieve one live birth. And the fact that intracellular cryoprotectant, but also the cold, were able to depolymerize microtubules put the focus on the possibility that the low outcome of oocyte slow cooling was connected to the possibility of an increasing aneuploidies in embryos generated by slow-cooled oocytes. But indeed, subsequent studies clearly demonstrated that, yes, it is true that the spindle, the metaphase to spindle in measured oocytes is completely depolymerized after thawing, but the microtubules repolymerize and correctly contact the metaphase to chromosomes. So other damage had to explain the low competence of embryos generated from slow-cooled oocytes in the low developmental competence of slow-cooled oocytes. And we did some studies on this. And indeed, slow cooling promotes a decrease in the number of cortical granules, and this was due to a premature exocytosis, and also there was a high fraction of the oocytes in which pericortical organelles were displaced toward the deeper cytoplasm. Moreover, we found in slow-cooled oocytes both normal mitochondria but also degenerated mitochondria. And using a mitochondrial inner membrane potential reporter that label in red highly polarized pericortical mitochondria, we also saw that this mitochondria, the potential of this mitochondria is lost after slow cooling and thawing, and this adds to other evidence shown by the group of Jonathan van Blercom, and he also suggested that mitochondria are essential engines in the developmental competence of the oocytes. So indeed, mitochondria, pericortical mitochondria, are needed to regulate calcium oscillation at fertilization. And in fact, at fertilization, we have a series of calcium oscillation, calcium transients, and after each transient, the intracellular calcium ions are recovered to basal levels thanks to the ATP produced by mitochondria and to the calcium ATPase of the calcium of the intracellular calcium store in the oocyte. And the sperm-triggered calcium oscillation regulate not only the exocytosis of cortical granules, but also the resumption of meiosis and the progression of the cell cycle. It also activates the translation of stored maternal transcripts, and the series of oscillation, a correct series of calcium oscillation is needed to have a correct pre and post implantation development. In the meantime, there were some papers by the Larman group showing that indeed all intracellular cryoprotectant that we use are able to elicit a large calcium transient. So both we have that propanediol, dimethyl sulfoxide, and ethylene glycol all are able to induce a premature calcium rise. And in those years, the paper by Kuwayama of the Kato ladies cleaning in Japan was published, in which he set a highly efficient vitrification method for cryopreservation of human oocytes that was successful in producing fairly good pregnancy rates. And so, as I told you, the vitrification is based on the brief exposure to high concentration of cryoprotectants. But what we have to keep in mind is that they may be toxic, especially at room temperature. So they are able to drive calcium increases, to cause osmotic shocks, and have also the potential to denature proteins, forming hydrogen bonds with proteins. It requires very high cooling rates in the order of 20,000, 30,000 degrees per minute, and the concept of minimum volume cooling. We can use open devices like the cryotop, and also closed devices that avoid the possibility of infectious agents transmission during cryopreservation or during the storage of different patient samples in the same nitrogen banks. And here you see a cryotip, a very thin device, a very thin pipette that promote very high cooling rates, but anyway, it is a bit tricky because it is very fragile and so may break during manipulation or during storage. And so we did another paper in which we studied the damage found in vitrified and slow frozen human oocytes. And we tested also our hypothesis that the damage to mitochondria found in slow-cooled oocytes may perturb the calcium oscillation at fertilization. We tested this by loading with the fluorescent calcium dyes both fresh, slow-cooled, and vitrified oocytes, and then challenging the oocytes with ionomycin to induce a large calcium transient. As you can see, here is the curve in terms of calcium excursion of fresh oocytes, and vitrified oocytes behave essentially in the same way. Whereas in the case of slow cooled oocytes, you can see that the recovery of calcium to basal levels is clearly compromised. You can appreciate this in this brief video, in which you can see the initial very low fluorescence of a slow cooled oocytes, and then you see the calcium rise induced by ionomycin, and the final level of fluorescence show that calcium is not recovered correctly. And the other damage, ultrastructural damage, we found in slow cooled oocytes were minimized in vitrified oocytes. So the formation of vacuoles and also the incidence of degenerated mitochondria and the organelle inward displacement that probably was due to perturbation of the cortical cytoskeleton. And on the basis of our findings and of the pertinent literature, in a recent review, we afforded an hypothesis on the causes of damage during oocyte cryopreservation. So, cryoprotectants like propanediol and ethylene glycol may passively veiculate calcium from the external medium, inducing a calcium increase in the oocyte cytoplasm. And dimethyl sulfoxide is able instead, according to the literature, to release calcium from internal calcium stores. And these in turn induce a premature cortical granule exocytosis, but this calcium overload may drive the opening of mitochondrial permeability transition pores, and this in turn may induce the release of calcium and of reactive oxygen species from the mitochondria, and the release of cytochrome C and various pro-apoptotic factors. So the survival of the towed oocytes depends on how much the mitochondrial pool is compromised. And so it can determine if the oocyte survive or dies. But survival is not everything. We have to determine the rest of the story of the oocyte, that is its developmental competence. And let's talk about female fertility preservation. It can be done through oocyte or ovarian tissue cryopreservation. Oocyte cryopreservation is offered to women with fertility-threatening diseases or treatments. For example, oncological patients that need to begin chemo or radiotherapy that can be extremely gonadotoxic. And it can be offered to women that wish to delay their reproduction for social reasons, so planned oocyte cryopreservation, or in the case of failure to obtain sperm on the day of oocyte pickup. And it is also indispensable for donor oocyte banking. So, although most studies suggest that vitrified oocytes perform equally compared to fresh oocytes, by fact, if you see this data from the US register, the outcome in terms of live birth rate per embryo transfer is lower for vitrified compared to fresh oocytes. And also it is possible that low quality oocytes or oocytes of aged patients may be more affected by vitrification, may be more damaged by vitrification. And one concept to consider is that during vitrification, the oocyte is exposed to osmolarities that are 10 to 20 times higher compared to the physiological osmolarity. And so the ability of the oocyte to withstand to the rapid fluid shift of water and cryoprotectant allow the oocyte to survive and perform well after vitrification warming. And just for that reason, some authors developed microfluidic devices to gradually expose the oocytes to increasing concentration of intracellular and extracellular cryoprotectants. And you can see a more regular shape of the oocytes using these devices compared to the manual exposure to abrupt changes in intracellular and extracellular cryoprotectant concentrations. And other means to improve our cryopreservation method is to increase the cooling rates, and this would allow the use of lower concentration of our toxic intracellular cryoprotectants. So this has been done using very thin and small, highly conductive quartz microcapillaries, and in this way cooling rates are accelerated to 200,000 degrees per minute. Or we can use another strategy, supercooled nitrogen, that is also called slush nitrogen. So when you dip a hot device in liquid nitrogen, gas bubbles develop, and this vapor layer delays the heat transfer between the hot device and the cold liquid nitrogen, and so it decreases the cooling rate. But instead, with supercooled nitrogen, you can see that the gas bubble does not develop, and so this effect that is called Leidenfrost effect is avoided, and we can reach higher cooling rates. But let's talk a bit more about the possibility of infectious transmissions to our samples. The fact is that liquid nitrogen is not sterile, and also the storage of patient samples using open devices in the same bank can lead to cross-contamination. But the fundamental question is, can closed system allow a sufficiently rapid cooling rate? And here you can see the use of a particular closed device in this brief video. So you see that the sample is loaded in a small well. The excess solution is recovered, and the oocyte is just covered by a film or medium to allow minimum volume cooling. In the meantime, an external sheet was plunged in liquid nitrogen, and the device is plunged into the sheet and then is slided into the liquid nitrogen. And according to the literature, open devices seem to maintain the performances of vitrified oocytes, embryos, and also ovarian tissue. But another mean to preserve the fertility, female fertility, is through collection of cortical ovarian tissues biopsies. And this can be offered to prepuberal patients or oncological patients or postpuberal patients in which tumor are estrogen responsive or they need to afford immediately the cancer therapy. And so the cortex, the cortical tissue can be cryopreserved and reimplanted, or can be cryopreserved and used, for example, for in vitro follicle growth. And the first births through ovarian tissue cryopreservation and transplantation were achieved years ago in 2004 and 2005 by Donnet and Meyrov. And the pregnancy rate were pretty fair, from 30 to 50%. And in 2021, Dolmans reported over 200 live births. And only 5 of these births were achieved after vitrification. But this does not mean that vitrification is not suitable for ovarian tissue cryopreservation, but the clinicians were more confident with the use of the slow-cooled method, so it was just a conservative thought. And the recovery of ovarian function occurred after 4 or 5 months or later in 95% of the patients. What can be improved is the duration. The duration of the recovery of ovarian function ranged from few months to years, but just in one patient it was registered a survival of ovarian function for 7 years. And this is due to the fact that up to 80% of the primordial follicles can be lost during the procedure. The more substantial loss is attributed to the post-grafting loss due to ischemic damage at transplantation, but also graft preparation and cryopreservation have the potential to diminish the ovarian reserve and so shorten the duration of the recovery of ovarian function. And as regards the vitrification and slow cooling of ovarian tissue, we have no sufficient clinical studies for stating which is better, but there are some research studies, and indeed the oocytes seems to be equally well preserved in follicles by slow cooling and vitrification of ovarian tissue, but some data suggest that the stromal cells that are sparse in the extracellular matrix of ovarian tissue have a high survival after vitrification. And we worked on human ovarian tissue vitrification using a protocol originally developed by Amorim for vitrifying human ovarian tissue in liquid nitrogen, and compare this procedure using liquid or slush nitrogen. And what we have to consider when we talk about the cryopreservation of a tissue, and so in the case of ovarian tissue, is that what is desirable during slow cooling of cells like sperm, oocyte, or embryos, that is, the formation of extracellular ice, may be dangerous in the case of tissue, because the extracellular matrix of the tissue can be damaged, and also the cells sparse in the extracellular matrix. And another factor to consider is that a tissue like ovarian tissue includes different cell types like oocytes and granulosa cells at various stages of development, and also stromal and thecal cells. And each of these cell type may have a preferred rate of cooling and susceptibility to cryoprotectant toxicity. And here you can see confocal images of fresh liquid nitrogen vitrified and slush nitrogen vitrified human ovarian tissue. And in blue, you can see the nuclei, and in red, the live dead far-red probe that stains both extracellular structure and dead cells. So you can see here a follicle with the dead oocyte in liquid nitrogen vitrified samples, and in fascia you can see the stromal cells that did not survive to vitrification warming with liquid nitrogen. So although follicles are equally preserved after liquid and slush nitrogen vitrification, we registered a lower survival of granulosa and stromal cells in liquid nitrogen vitrified samples. And you can see this in these electron microscopic images of granulosa cells, damaged granulosa cells, and stromal cells in liquid nitrogen vitrified samples compared to intact follicle cells and stromal cells in slush nitrogen vitrified samples. And we also examined a panel of genes involved in crescent toxicity pathway, and the genes were more dysregulated compared to fresh oocytes in liquid nitrogen vitrified oocytes. But as I told you, the survival at thawing or warming, of course, is important, but this does not mean that the cell perform correctly. And in fact, if we culture the warmed liquid nitrogen / nitrogen vitrified human ovarian tissue for 9 days, you can see that the primordial follicles present in fresh tissue are able to activate and progress through the primary and secondary stage equally well in liquid nitrogen and / nitrogen vitrified tissues. But if you consider the follicle survival at 9 days, you can see a 50% survival in the case of liquid nitrogen vitrified tissues, opposed to 80% survival in the case of / nitrogen vitrified tissues. And so it is important to continue to investigate on the cryopreservation of human ovarian tissue, because the cryopreservation itself may deplete the ovarian reserve, and this is fundamental to try to extend the lifespan of the graft after reimplantation. And so, in conclusion, I hope to have shown you that cryopreservation is an indispensable adjunct in assisted reproductive technique, and the main strategies used currently are equilibrium cooling and vitrification. Slow cooling is the first procedure developed, but it is being gradually replaced by vitrification. Vitrification, since is a manual procedure, we have to take in mind that we think to have applied a vitrification procedure. This does not mean that we correctly vitrified the sample, and so we can cause by manipulating our devices through the cryoprotectant concentration, a too long exposure or a too short exposure to intracellular cryoprotectant, or too low cooling rate, and so this is very highly dependent on the operator skill. And more studies are needed to understand the effect of the particular cryopreservation procedure on ovarian tissue cryopreservation and transplantation. Thank you for your attention.